DNA replication. Humans are made up of cells. In our cells, we have DNA. We humans start off as a cell with DNA that keeps dividing. These cells and DNA inside divide because of mitosis, meiosis, and more. When these cells divide, it requires information, aka DNA inside of these cells, to be copied, and that is what DNA replication does. DNA replication is like a construction project. There are many different jobs that are required. Think of it as a zipper on your jacket. The zipper on your jacket represents a double helix molecule. One side of your zipper represents one strand of DNA and the other represents the other strand of DNA. Let the slider represent the helicase. The helicase is a divider. It divides the two connected strands of DNA into their own separate strands. Now we have two separate strands. But the problem is that these strands are alone, and they need a new replicated strand to make a double helix molecule. This is where the primase comes in. The primase builds a primer, and the primer is where the DNA polymerase comes in, aka the builder. It builds on from the primer, continuously adding new DNA bases to make a new strand. Now we've successfully understood what happens with one DNA strand, but what about the other one? You may think that the other strand of DNA that was unzipped may go through the same process, but it actually does not. DNA strands are anti-parallel. That means that DNA strands are parallel, but they move in opposite directions. So one DNA strand starts at 5 and ends at 3, while its other strand starts at 3 and ends at 5. There is a reason behind why we use 5 and 3, and that's because of carbon atoms that make up DNA. But anyways, that means one strand of the unzipped DNA will be a leading strand, so the polymerase can continuously build DNA bases to create the new strand. But that also means that the other strand of the unzipped DNA will have a lagging strand, not being able to continuously build DNA bases. So for this lagging strand, it still goes through the processes as the leading strand, but since it can't build continuously, the polymerase rather builds in chunks called Okazaki fragments from the primers. And so that is DNA replication.